Part 6 in our electrophysiology lecture series. We're getting to the point where we've covered two main topics now, and we're going to start putting them together. So we've covered the depolarization of cells, which is the excitation of the cells, and we've talked about the contraction of the cells with the actin and the myosin. So now we're going to connect those two dots and talk about how that action potential that we've been talking about starts to lead to the physical contraction of the heart. Because of course, sometimes you can have the action potential firing off, but there's no actual contraction. There's such a thing as pulseless electrical activity, where we have electrical activity in the heart, but no contraction. So how do we make those two connect? If you go to paramedicine.com, there's a URL link there to slideshare.net. You can download the PowerPoint for these slides, and you can use those PowerPoints as you wish, according to the Creative Commons license. This is the 6th lecture in an 8-part series, so be sure to look for the other ones. If you're watching on YouTube, there should be a link to all the other lectures at the bottom of the description, and if you're on paramedicine.com, then you can just see them all on the paramedicine.com page. Some priming questions to get you ready, questions you should be able to answer. And I realized in the previous lecture, I didn't answer one of the questions. And the question that I had as a priming question was, what do we mean when we say functional syncytium? So I put the answer in the comments below the actual video itself. Sorry about that. So here are the priming questions for us here. What are the 10 stages of excitation contraction coupling? What ion is the driver of contraction? And you should know that from the last lecture. The answer is calcium. And we're going to put a diagram up on the following slide. And basically, I'm going to talk through this diagram in 10 steps, and you'll get an idea of how this all works. So here's the diagram. I'll leave it up here. Again, if you want to get this raw diagram with nothing labeled on it, with no information in it like this, you can get it off the slide share. So just download the PowerPoint that I'm using, and you'll be able to get this slide. So here's the question. We're depolarizing myocardial cells. How do we then get them to cause... Sorry, I got a dog here and he's looking at my foot. Easy, Ralph. How do we get them to start the contraction, the actin-myosin contraction inside? Just because the plasma membrane is depolarizing doesn't necessarily mean anything's going on deep inside the cell. So how do we get that to work? That process of connecting the two is the excitation contraction coupling. So by review, as way of review, there's 3 ways that we can depolarize a myocyte. That's not make it contracting, but just make it fire off electrically, get the ions moving across the plasma membrane. The first one is in a pacemaker cell, which we discussed in a separate lecture, that they have automaticity through their funny channels that make them fire off spontaneously at set rate. That's condition number 1. Condition number 2 is maybe you've got 2 myocytes together, and when one myocyte fires off, we get that wave of depolarization going along the membrane. That wave continues through special junctions in the gap between, called gap junctions, that allow the ions to fire through, and that sets off the resting myocyte, so it starts to depolarize. And the other thing, the third way we can do this is we have Purkinje fibers that innervate through the ventricles, and the Purkinje fibers carry a wave of depolarization, and so if a resting myocyte is attached to one of these Purkinje fibers, and the Purkinje fiber is depolarizing, then the Purkinje fiber will make the resting myocyte depolarize. So we've got depolarization happening. That's how it happens. Depolarization happens mostly at the membrane level. So how do we get that electrical exchange of sodium and potassium and a bit of calcium to go deep into the cell to cause the actin and myosin to start their contraction, the power stroke? That's the question. Let's take a look at this cell. All the things are filled in now. So here, the white is the outside of the cell. The yellow is the inside of the cell. Just ignore all the stuff that's in there for now. And you'll see we've got extracellular fluid and intracellular fluid here. And this is a wave, this bit here, is a wave of depolarization going across the plasma membrane of the cell. Somehow we've got to get that down into the sarcoplasmic reticulum so that it releases calcium, which is what we said causes the actin and the myosin to do the power stroke and contract. So as this wave of depolarization passes along the surface, it goes down through the T tubules, the transverse tubules, which go deep into the cell. And then the current continues to go up, around, and then continue across. But as it comes down through the T tubules, there are these channels in the T tubules, which are voltage gated channels. So as this wave of depolarization comes down here, it hits these channels and it opens them up. And these are long-lasting calcium channels. So there's a bit of calcium floating around in the extracellular fluid. And when these channels, these calcium channels open up, the calcium comes in to the cell. That's step 2. So we're on step 1 here. The action potential comes along and goes down into the T tubule. And I've jumped into step 2 a little bit, and that is this voltage gated channel then opens and calcium comes through the plasma membrane and into the intracellular fluid inside the cell. This channel, by the way, is where some calcium channel blockers work. So to apply this a little bit clinically, some calcium channel blockers will work on the blood vessels throughout the body. And by blocking calcium channels there, they make it difficult for the blood vessels to constrict, because blood vessels use actin and myosin as well. Those are the muscles that, or the fibers that cause those muscles to contract to make the vessel smaller. So by using a peripherally acting calcium channel blocker, you can stop a lot of calcium going in, so the muscles can't contract as much, so they're more dilated, you're more vasodilated, and we can drop people's blood pressure. So if they have chronically high blood pressure, then that's one of the ways we can drop them. And you can tell if people are on calcium channel blockers. Sometimes they go into a first-degree block, but also if you take a look at their ankles, because they're vasodilated, they get dependent edema, and they look like somebody who's got congestive heart failure, even though they don't. So if you see somebody whose socks are cutting in, you pull down their socks and there's the impression in there, you can ask if they're on calcium channel blockers. Other calcium channel blockers work in the heart, and they work in particular on this membrane. So verapamil is more of a cardioselective channel blocker. Its trade name is isoptin. You might know it by that. And isoptin can block this. Now, since this is the beginning of this whole series of things which makes the heart contract, you can imagine that if we give someone a cardioselective or more cardioselective, works more in the heart than the blood vessels, calcium channel blocker like verapamil, you're going to slow down or decrease the amount of squeeze. You're going to have the calcium channel blocker working as a negative inotrope. So the heart can't squeeze as strongly. So calcium comes in, as long as you're not on a whole bunch of verapamil, comes in and goes into the sarcoplasmic reticulum in the cell, and in particular the deep cisternae. And inside those deep cisternae in the sarcoplasmic reticulum is a whole bunch of calcium. When I'm explaining this in class, I usually use the comparison that sarcoplasmic reticulum is like, you know how people go climbing, rock climbing or wall climbing in a gym, and they have a bag full of chalk? I think of the sarcoplasmic reticulum like that. It's just this bag full of chalk. And so what happens is, because it's kind of a weird bag, when a bit of calcium hits the bag, it just goes, and this big cloud of calcium comes out. It kind of goes and puffs out all the calcium into the intracellular fluid. So we get kind of like a calcium explosion. And that happens because the calcium comes here through the voltage-gated channel, through that calcium channel, hits the sarcoplasmic reticulum, and induces the release of a whole bunch of more calcium. So a little bit of calcium stimulates a lot of calcium, and we call this calcium-induced calcium release, which is a term you might read a little bit when you're reading about electrophysiology. So this first step, the depolarization, the opening of the calcium channel, the calcium entering, hitting the sarcoplasmic reticulum, and poof, releasing a whole bunch of calcium, is the first step, which we call calcium-induced calcium release. Now, if you want to dive a little bit deeper here, this is geek stuff, but I fully encourage you to geek out. These receptors here are called ryanodine receptors because there's a drug called ryanodine that will bind to them. So it's a convenient name to call that. And similarly, these long type calcium channel voltage-gated channels are called dihydropyridine channels or receptors, because the hydropyridine is a drug that will bind to them experimentally. So yeah, you don't need to know that, but if you're kind of keen, you like to geek out, then go ahead, go deep. Okay, so the local release of calcium here from the sarcoplasmic reticulum causes this puff or cloud of calcium to come out into the intracellular fluid and make its way down to where the myosin and the myosin heads are attached to the actin. So all of these sparks create a calcium channel, a calcium flow, which travels down to the actin and myosin. And of course, when they hit there, they do what we talked about in the last little lecture in this series. That is, the calcium hits the troponin C, which stands for calcium, and it takes the tropomyosin off of the actin, and that allows the motor heads of the myosin to grab along the actin until the troponin I kicks the calcium off, and then the I for inactivation. The tropomyosin comes back over the actin, and it causes relaxation inside the muscle. So we power stroke, and then tropomyosin comes back, and then we release, and the muscle is reset, ready to go again. Now, the actin and myosin are reset down here in stage 7, but we still got a whole bunch of calcium floating around. And so we've got to get rid of that calcium and put it back where it belongs to get ready for the next act when we do the whole thing again, 60 to a few hundred times a minute. So roughly once a second, our heart is doing this. So we've got to reset now. How do we reset? Well, the calcium from down here is actively pumped back into the sarcoplasmic reticulum by an adenosine triphosphate pump. So good old ATP, battery of the body, is powering pumps on the walls of the sarcoplasmic reticulum, which sequester calcium back into the sarcoplasmic reticulum. It's basically going and sucking up all the chalk dust and pushing it back into the bag on the belt of the cell so that the calcium is ready to puff again and stimulate the actin and myosin to contract. But we still got some more calcium floating around that we need to get out of the cell so that that calcium can be put back into the cell for the calcium-induced calcium release. We get that out of the cell by it comes close to the plasma membrane here. And you'll remember, hopefully, from a previous lecture, that we've got the sodium and calcium exchanger here. This doesn't use adenosine. When we did this diagram before, which I love, we had the two exchangers up here, which use ATP in order to power mechanically the exchange of ions. And that creates a concentration gradient, which these two exchangers work off of. And this exchanger in particular is the sodium-calcium exchanger. So we've got all this sodium, or calcium rather, floating around inside the intracellular fluid. We want to get it out. So we get up to this exchanger, and it basically works like one of those round glass revolving doors. We stick 3 calcium on the inside part of the door and 2 sodium on the outside part of the door, and when they fill up, they just magically switch around. The calcium gets spit out of the cell, the sodium goes into the cell. So now we've got the calcium out. But we got a problem because now we have a whole bunch of sodium in. And in its resting state, we have sodium outside the cell and potassium inside the cell, but we just threw 3 more sodium into the cell, so we got to get them out. So the way we get them out, next slide, is this sodium here finds its way over to a sodium-potassium pump, which is powered by ATP. And we kick the 3 sodiums out, and the potassium, which is floating around out there, gets kicked back in. So that's a really beautiful, elegant way that our bodies take this action potential, which is happening outside the cell, thrust it deep into the walls of the cell through the transverse tubule, a little bit of calcium-induced calcium release. That calcium hits the troponin C, which takes the tropomyosin off the actin. We get a contraction. The troponin I kicks the calcium off. We get relaxation. Some calcium goes into the sarcoplasmic reticulum. Some gets kicked out of the cell. In the process, we get some sodium pushed back into the cell, but that's okay because we can push it mechanically out of the cell using a little bit of adenosine triphosphate. That's why you need to eat your sugar and breathe your oxygen, because without that, you can't recharge your adenosine triphosphate. Sodium goes out, potassium comes in, we're ready to go again. Beautiful, beautiful, fantastic, complex system. So you'll remember again, sodium-potassium pump, we talked about that on the previous model. That's the main pump that we have in our cells for exchanging ions. So just to sum up, we've got excitation and we've got contraction. Excitation is this action potential in green. So sodium rushing in potassium, then a little bit of calcium, and then everything gradually resets. If we just have that happening, there's no squeeze. But as we go through here and we have a little bit of calcium come into the cell to extend this plateau, it changes the charge, which extends the plateau, but it also stimulates the calcium-induced calcium release. So we have calcium here now flooding over the troponin C, and from here, oh, where'd my mouse go? There we go. From here, we start to get our actual physical contraction. So this is the tension in the heart wall. This is the squeeze that the heart's going through, and we get a really good strong squeeze, and by the end of the action potential, as it's reset, our actin and myosin fibrils have reset as well, and within about 300 milliseconds, we've depolarized, contracted, repolarized, and released the heart, and it's ready to go again. Beautiful, just a beautiful system. So that's how it all comes together. That's one big chunk connecting with another big chunk, and that explains how we can take an electrical impulse, exchange of ions, which is happening way up on the plasma membrane, bring it deep down into the cell to allow the actin and myosin filaments to slide over each other to actually cause physical mechanical contraction. And of course, when you mechanically contract a sphere, an orb, it squeezes what's ever inside. So if there's a hole in there, it gets squeezed out, and that's how the heart does its pumping, because you can only squeeze in one direction because the valves stop the blood from flowing back. So when you squeeze it, it goes always in the appropriate anatomical, physiological direction, as long as the heart's working properly. Cool, eh? So that's putting two big pieces together. It's always fun when I do this in the lecture, in the actual live lecture with students, because you see people thinking as they go through, and then I explain this bit and they go, oh, it's kind of fun. If you have any questions, feel free to put them below in the YouTube. I do check these regularly, as you'll see, or you can email us, contact@paramedicine.com. Thanks a lot for watching.